mariculture. So in this, we are going to discuss the objectives, introduction, tiger shrimp culture, oyster culture, pearl culture, seaweed culture. Now, what are the objectives of this chapter? After studying this unit, you will be able to learn the importance of shrimp culture to meet food demand. You would understand oyster culture, how it is done, and you would also learn the pearl formation, how the pearl culturing is done, and we'll know about seaweed culture also. Now let's get into the topic. Brackish water prawn and fish culture is traditionally practiced in West Bengal and Kerala. Traditional system is based upon trapping the natural seed, holding and fattening them in impounded tidal swamps and coastal paddy fields. Major defects in traditional culture and the large-scale wastage of juvenile stocks of desired species are uncontrolled species and composition and density, and as low as unpredictable production. Researchers carried out since last decade have laid the foundation of scientific brackish water aquaculture technology and enhanced the yield potential to an appreciable extent. Since the blue revolution has taken place, people are concentrating more on production of the fish. So whether it is in a natural state or culture farming. Now, what are the rules and regulations? What are the precautions to be taken while having culture farming? Now, prawns are the primary cultivated organisms in coastal aquaculture system. Brackish water farming in India at present depends on the stocking material available in nature. Observation on seasonal availability, abundance, and suitable gears for collection of prawns and fish seed have been carried out in different brackish water areas situated along the east and west coast of India, including Godavari Estuary, Kakinada and Pulicat Lake of Andhra Pradesh. Controlled breeding of cultivated prawns and hatchery production of their seed is a recent development in India. Let us learn about tiger shrimp culture. Peneus monodon, the scientific name is Peneus monodon, and the tiger prawn, it is the largest growing species among the penaeid prawns. It is abundantly available in all maritime states on the east coast. Culture of tiger prawn is a known practice traditionally done in Pokali fields in Kerala and Beharis in West Bengal. In several other coastal states also, tiger prawn is cultured in brackish water forms. Besides government agencies, a number of private entrepreneurs have started growing this prawn as it has a tremendous demand in national and international markets. About 207 million hectare brackish waterland is available in our country for prawn culture, but a little area is under cultivation. Now see, pond preparation. It is important to ensure better production by eradicating predatory and unwanted organisms to generate natural productivity and growth of prawns. It involves site selection, drying, liming, watering, and manuring of pond. Let us see each one by one. What is site selection? The site should be free from mangrove vegetation and close to brackish water areas. Sandy areas should be avoided. The area gets inundated during high tide. Ensure that high flood levels are not very high and it should not be more than 2.5 meters above the ground level. Select the site which is gently sloping and fairly flat. The soil should be clay or loam clay, and soil pH should not be acidic. For better growth of prawns, the water should have temperature from 26 to 30 degrees centigrade, 32 degrees centigrade. Salinity should be between 14 and 25. And pH should be in between 7.5 to 8.8. Dissolved oxygen should be more than 4 ppm and iron should be less than 2 ppm. The site should have freshwater facilities in adequate quantities. Access by road should be there. The site should have electricity facilities. The site should be available in good quantities near to the site. Next is drying of pond. Before raising a crop of prawns, the pond should be thoroughly dewatered during low tide. Expose the bottom to the sun for drying for a few days. Plow the bottom thoroughly. This practice helps in the release of obnoxious gases, helps in mineralization, and ensures eradication of predatory and weedy fishes and other harmful organisms. Next, it is after drying of pond, we need to concentrate on liming. What is liming? Lime helps in killing pathogenic microorganisms. It controls pH fluctuations and to improve hygienic conditions of the pond bottom. Lime is applied at the rate of 250 kg per hectare before water is let into the pond. After liming then comes to watering. A few days after liming, tidal water from the creek is let into the ponds continuously during the high tide. The water inlets are grounded with nylon screens. It restricts the entry of predators and weed fishes into the pond and helps in higher survival of prawns. The water level should be maintained at desirable level. Next, manuring. It should be done with the tidal water and helps the substance for the prawns. Additional manures are required to produce prawn food organisms, which are most important for faster growth of prawns. These should attain marketable size within 3 months' time. To increase the nutrients and prawn food organisms, manuring of pond is most essential. Application of both organic and inorganic fertilizers 5 days earlier to stocking gives good bloom of plankton. 1000 kg per hectare crop raw cattle dung, 125 kgs per hectare crop single superphosphate, and 25 kgs per hectare crop urea are necessary to increase the productivity of pond. Half of the above dose of manures is applied to the pond 5 days before stocking, and the balance of the manure in 4 equal fortnight installments after stocking. These fertilizers are mixed thoroughly to prepare a semi-liquid paste in a tub and spread uniformly on the water surface. Next, coming to stocking and post-stocking pond management. It includes procurement of seed, acclimatization, stocking, water management, supplemental feeding, and harvesting. Now, what is procurement of seed? It depends on the collection of post-larvae and juveniles from tidal estuaries, creeks, and intertidal pits for stocking. Low-lying areas and mangrove swamps along the coastal areas are important nursery grounds for juveniles of prawns. These areas are always under tidal influence, and the seed enter into these areas alone with tidal water. Tiger prawn is euryhaline and migrates for feeding into these areas, which are rich with periphyton. During high tides, millions of juveniles can be congregating in these grounds. These seed can be collected by a simple method of trapping with hand nets and can be transported to the farm. Next is acclimatization. Care has to be taken to acclimatize the young prawns procured from natural resources to the pond condition before stocking. Prawn seed in its natural habitat thrive in a salinity which is lower than that of pond water. Therefore, direct release of the prawn seed in pond water results in high mortality. Hence, it's most essential that the seed brought from natural resources and hatcheries are acclimatized gradually to the salinity of the pond water. This ensures a higher survival rate. Next is stocking. The criteria for the selection of suitable prawn for culture are: number one, ecological adaptability; number two, availability of stocking materials; number three, ability to grow fast with available food; number four, ability to accept supplementary food; number five comes to market value. So, prawns that are naturally available in the locality will be the most suited for farming. Peneus monodon is the most desired species for farming due to its fast growth, hardness, tolerance during handling, adjustment to fluctuations in culture media, and fetching attractive price in the market. Next is water management. During the culture period, water level should be maintained at 60 to 80 centimeters. The creek water let into the pond carries rich nutrients and abundant plankton necessary for a good prawn growth. The continuous supply of water to pond enriched with nutrients for 5 to 7 days once in a month, it helps in replenishment of nutrients and in nitric plankton. Besides ensuring removal of metabolites and providing a aeration. This practice helps in rapid growth of prawns. Poor quality of water may cause cannibalism, bacterial and viral attack, and other infections. Periodical checking of sluice gates, screen, and the bun to plug the holes is necessary. Some of the water can also be exchanged during the culture period based on the day-to-day observation of the condition of the pond. Next, supplementary feeding. Prawns are carnivorous and bottom feeders. Prawns, periphyton, and detritus. As they grow, the demand for more food increases and they start eating the smaller prawns. Naturally occurring food organisms get depleted and hence they have to be supplemented with artificial feed during late evening and early morning hours. To obtain quick growth of prawns, the artificial feed is to be supplemented with the right proportion of protein, carbohydrate, fat, minerals, etc. in the form of pellets. Wet feed includes broken rice mixed with clam flesh, trash fish, frog waste, shrimp head, and mussel meat. Every day about 3 kgs broken rice during first 60 days and same quantity of broken rice along with 1 kg of flesh per day for next 30 days is to be supplied for 20,000 juveniles per hectare. This combination of food for 90 days results in the growth of prawns to a marketable size. It is better to use check trays for feeding as these are useful to observe the feed consumption. Sampling should be done for monitoring growth and survival once in every 10 or 15 days with cast net so as to assess the growth and survival of the prawns. Examine the prawns for any symptoms of diseases, external injuries and malformations, feeding conditions, and assess the biomass in the pond also. Next, coming to harvesting and marketing. The growth and survival of prawns depend upon several factors such as species stocked, culture system adopted, and managerial methods followed. Tiger prawns can be harvested after a culture period of 3 to 4 months when it reaches 40 to 50 gram weight. Harvesting can be done by draining the pond water through hag net placed in the sluice. Harvesting can also be done with cast nets, and the leftover prawns can be hand picked after draining the water. Then, harvested prawns need to be kept in layers of crushed ice before marketing. Next, what are the different stages of prawn farming? Based on the management adopted in different parts of the world, prawn farming can be termed as traditional, extensive, semi-intensive, and intensive. Then, culture of tiger prawns in freshwater. These are also cultured in freshwater ponds with saline soils. It is necessary that the soil of the freshwater ponds should be saline in nature, though the water may not have salinity. The results indicate that the survival, growth, and production in freshwater ponds is almost similar to that of the culture in brackish water. Now let us learn about the oyster culture. Large quantities of the edible oysters, that is Crassostrea madresensis, growing widely in most of the tidal creeks and estuarine regions along the east coast of India. The shell time industry, however, quarry agglutinated dead shells from rugged beds and those found as subfossil deposits in riverbeds. Realizing the edibility of the oyster meat, Hornell initiated oyster culture experiments early as in 1910 at Pulicat Lake. As a part of global strategy for developing oyster farming, following the great strides made in this venture by developed nations, the Central Marine Fisheries Research Institute focused its attention on developing systems for the culture of edible bivalves, identifying edible oyster farming and mussel farming and priority areas for research and development. Evaluation of the resources potential, identification of suitable water spread, and areas for culture evolving proper techniques to collect required seed for farming, introducing an appropriate method of farming, and establishing a model farm formed the broad objectives of the project initiated in 1975 on edible oyster culture. Now, this has various types. Number one is spat collection. Of the several types of spat collector like bamboo reapers, concrete blocks, nylon meshed net pieces, bamboo mats, strings of coconut shells, shells of green mussel, and oyster shell walls, and country tiles tried in different locations around the bed, the most satisfactory results were obtained from twice lime coated semi cylindrical country tiles laid completely submerged on wooden platforms. Determining the accurately the spawning period of oysters and laying tiles just at the right time of release of oyster spawn increases the percentage of success. April to May spawning period proved to be the ideal time for large scale collection work. Post spat collection period. Spat removal. Spat settled on collectors were allowed to grow on them up to a size of 30 to 35 millimeters, after which removal of individual spat is easily done without injuring the fleshy interior. Pressure slightly exerted dislodges the spat without damage. After the removal of spat, the titles can be stored and recycled for use in the next season. Depending on handling, they are good for at least 4 seasons. Initial transplantation. Initially, the scrapped oysterlings are put in nylon meshed cages of 12 mesh size of 6 iron rod frame for a period of 2 months. Each cage measuring 40 into 40 into 10 centimeters can be easily hold 200 oysterlings. Later, they can be transferred to large rectangular cages of 22 meshed nylon netting, sizing 90 into 60 into 15 centimeters. Erecting racks for growing oysters. Each rack is so constructed as to occupy an area of 26.5 square meters with a length of 13.2 meters and breadth of 2 meters. A mid-water wooden platform of inner connected teak wood stubs of 2 meter length is put up supported by two parallel rows of 6 teak wood poles, each planted vertically down at a distance of 2 meters, pole to pole. All wooden materials are treated with tar prior to being used in the rack erection. The platform in each rack can carry a total of 20 rectangular cages of 150 to 200 oysters and is so positioned that only during the lowest low tide the cages get partially exposed. The oyster culture experiment successfully carried out at Tuticorin marks only the first stage in an era of oyster industry in India. Based on the experiences during the course of these experiments, several areas of research and development efforts appear to be warranted on prior basis before a total success can be achieved. Spat collection is one aspect where the present experiments have indicated the enormous potentialities of the coastal regions, which can be usefully tapped to gel adequate guide supply from natural resources. Although only a few of spat were collected for the experiments at Tuticorin, it appears distinctly possible to collect several millions by employing a large number of spat collectors using the lime-coated tile technique. The rack culture system yielded fruitful results in harvestable oysters being made available within a short span of 12 months, unlike in other countries where growing period is protracted. Production of an average of 0.48 tons of oysters from a single rack has been demonstrated at Tuticorin. In 1 hectare, it is possible to put up at least 280 racks and stock nearly 1 million oysters, resulting in an yield of minimum 135 tons of oysters with shell, with a total net yield of 13.5 tons. Experiments are in progress to evolve modifications towards bringing down the capital investment on materials. Future research in this direction would aim at employing less costly material for rack erection as well as for the trays used for growing oysters. Next, coming to pearl culture. Blue Revolution not only includes production of food, but also culture of other than food items like pearls, which are export value. The beauty of pearl is known to mankind since 3500 BC, according to an old Chinese scribe. The pearl is one among us the first game to Adam mankind. Even the Vedas in India, the Bible and the Quran made several references to pearl as a symbol of purity, beauty, health, and wealth. Now, what are these pearls? How they are prepared? How they are manufactured? Pearl is one of the most precious, richest, and most beautiful of human ornament and is classed with diamonds. Pearl is one among the 9 gems. A pearl merely consists of concentric layers of nacre or matter of pearl around a nucleus. Nacre or innermost layer of the shell is composed of a series of thick plates or laminae overlapping one another, arranged parallel to the shell's surface. The edges of these laminae are zigzag, and since the surface shows both faces and edges of the laminae, this irregular surface displays the wonderful and so greatly admired iridences. Haliotis and piano trocars have bright nacre and are much used in the manufacture of buttons, etc. Pearls are most lustrous than mother of pearls because the nacre is deposited in exceedingly thin layers. These laminae overlap one another. The surface appear crumpled and corrugated than viewed through a microscope. A pearl merely consists of concentric layers of nacre or matter of pearl around a nucleus. Nacre or the innermost layers of the shell is composed of a series of thick plates or laminae overlapping one another and are arranged parallel to the shell surface. Look at the pearls, how beautiful they look. The shiny and lustrous beauty is admirable. The greater the angle which the laminae from with the surface, the closer will be these serrated outlines. And where the plane of exterior laminae is parallel with that of surface, the lines are not present. This deposition causes the waves of light to be reflected from different levels of the surface, just as in soap bubble, and the minute prisms split up the rays into their colored constituents, producing the iridescence. Pearl consists of 91.72% of calcium carbonate, 5.94% of organic matter, and 2.34% of water. Strands of pearls should never be dipped in water or solution. They should be cleaned with a cloth. The large, exceptionally beautiful pearls are called paragons. Now, what is the history behind this origin of pearls? Many theories were put forward to explain the origin of pearls. One theory stated that the pearl was gallstone of the oyster. Another theory said When surplus fluid developed by the oyster but not discharged remained, the condensed in the form of and condensed in the form of a pearl. The theory which gained more popularity is that sand grains get into the shell to form pearls, and parasites or their eggs or other organic matter form the core of the pearl. In 1907, Tokichi Nishikawa gave the most possible scientific explanation on the origin of pearl. His theory later came to be known as the pearl sac theory, which explains that a pearl is formed when the pearl secreting cells of the mantle migrate into the body of the oyster under the stimulus of a foreign body and form a pearl sac by cell division around the foreign body. The pearl sac secretes nacre, which gets deposited on the foreign body in layers, and in course of time, a pearl is formed. Let's see the types of pearls. Based on the shapes of pearls, there are four types: regular pearls, irregular pearls, semicircular pearls, and reflectory pearls. Regular pearls, these are 3 to 4 mm in size with silvery pink color. These will be produced by grafting the nucleus into gonads of mussels. It is most common in Japan. Then irregular pearls. These are without nucleus, irregular and small, about 2 mm in size. These are prepared around the mantle graft in mantle tissue of a mussel. China produces more number of irregular pearls. Next, semicircular pearls. These are either in silver or gold or pinkish in color and grow to 5 to 8 mm. These are produced in mussel mantle where the mucus is grafted. Next, the last one is reflectory pearls. These are very large, about 10 mm in size, and are in different shapes. These are produced in mantle of mussel when selected shaped foreign body in introduced. Pearl producing mollusks. Freshwater pearl producing mussels belong to the family Unio- Unioidae and are considerably important for production of pearls all over the world. These are Heliopsis sklegish in Japan and Chrysaria plicat in China. Quadrula species, Pleurobimus species, Tritogonia species, Megamias in USA. Unio species, Margitifera species in Europe, Pariaceae corrugata in Bangladesh, Lamellidens marginals, Lamellidens coreanus, Lamellidens corrugata in India, and Heliopsis mingi in Vietnam. Freshwater pearl culture in the world and in India. The quantum of pearl exploited from natural sources are too meager to fulfill the demand of the people. The shapes and sizes of natural pearls are not uniform in nature, which creates sense of dissatisfaction in consumers. This stimulation led the people to think of an assured source of quality pearl supply. Chinese were the first to initiate pearl culture in freshwater mussels in the 12th century. In Japan, the freshwater pearl culture commenced in 1935 in Lake Biwa using an indigenous mussel species, Heliopsis sklegilli. In between 1967 and 77, Japan produced 4 to 7 tons of pearls. Chinese started pearl culture practice on a large scale in 1960 and produced 50 to 80 tons per year by using improved technology. China led the world in freshwater pearl production since 1985. Freshwater pearl culture. Methods of freshwater pearl culture vary depending on internal structure of pearl mussels and the level of expertise available. There are three important techniques which are in vogue: mantle cavity insertion, mantle tissue insertion, gonadal insertion. Mantle cavity insertion. This technique is followed generally to produce pearl images and half round pearls. For this purpose, muscles of 6 centimeters above are carefully opened 1 centimeter wide without causing injury to their soft inner parts. The walls are kept open by means of a wooden wedge. A small area of the mantle is detached from the upper shell wall, and 2 to 4 shell beads, that is up to 6 to 10 diameter, are pushed into the interior, middle, and posterior part of the mantle cavity. Similar implantations are made on the opposite shell wall also. Then mantle tissue insertion. By this method, non-nucleated pearl of free irregular size and small round pearls can be produced. The technique involves the following steps: collection of muscle and pre-culture. Mussels having 10 to 15 centimeter shell length are collected and sorted out into two groups. The mussel to be operated upon, termed as operation mussels, and those to be sacrificed, designated as cell mussel. These mussels are kept in a basket, 215 per basket, and suspended in lake, 3 to 5 per basket. The period of pre-operative culture varies from 1 to 3 years. Next, surgery. After taking out the mother mussel into the fresh water basin, it is opened up by cutting that muscles. The pallial mantle of the cell muscle is cut into 0.5 centimeter wide and 7 centimeter long stripe. The strip is then sliced into a number of small square pieces up to 0.5 centimeter square. Thereafter, using a shell opener, the valves are opened and a wooden wedge inserted at the edge of the shell to keep it open while inserting the cut square piece of the pallial mantle. A number of small holes without piercing through the mantle are made on the exposed surface of mantle by means of a specialized needle. Then the cut live mantle pieces collected from the cell muscle are inserted in these holes, with the outer side of the graft facing the inner side of the operation muscle shells. Then the grafting is done on both sides of the mantle. The operation muscle of 10 centimeters length can receive 20 to 40 implants, both sides combined. In a nucleated operation, a nucleus is placed in the hole along with the mantle, grafting in close contact for obtaining round pearls. Post-operative culture. The operated muscles immediately after implantation are placed in a tub of clean fresh water in a vertical slide up position and put in the culture pond. 10 to 12 operated muscles. This is retained in water for 3 years with periodic cleaning and maintenance of the unit. Then coming to harvesting. At the end of culture period, pearls are harvested either by crushing shell or by taking out individual pearls from the pearl sac of the mussel. Then coming to gonadal insertion. Gonadal implantation of graft and nucleus being practiced in the marine environment for pearl oyster. Recently, Japan has successfully employed this method for freshwater pearl culture production. By this method, regular round oval larger culture pearls of 6 mm diameter can be obtained. It has three steps. These are: step 1, procurement of live mussel and pre-operative conditioning. Prior to operation, mussels of 8 centimeters in length and 50 grams in weight or above are sorted out and are kept in a freshwater tank, which is having 200 liter to 500 liter tank capacity for 5 to 7 days. Crowding of muscles in this way for shorter period ensures proper relaxation of abductor muscle in preparation for surgery. Next, step 2, implantation of graft and nucleus. From a donor muscle, pallial mantle is obtained, cut into a number of pieces, and kept moist till implantation. Having prepared all these, the recipient muscles are subjected to gonad implantation, making use of tools. The graft is slowly inserted deep through the incision, making sure that the outer side of the graft is facing the entry point. Water quality management. Pond culture period of operated mussel depends on the fertility of water. Therefore, it is desirable to fertilize the ponds in split monthly doses, either with organic manure, that is 10,000 kg per hectare per year, or with a chemical fertilizer like urea, 100 kg per hectare per year, to maintain good algal density in the culture waters. To avoid pollution effect, adjoining ponds can be fertilized and algal-rich water taken into pearl culture pond. The desirable phytoplankton organism includes Chlorococcum species, Pediastrum species, diatoms, etc. These algal forms constitute natural food organisms for the mussels and provide essential nutrients like calcium and silica for biomineralization of pearl nacre over implanted nuclei. Expensive blooms of microcystis species and euglena species are to be avoided in pearl culture water, obtaining quality pearls, for obtaining quality pearls. Next, marine pearl culture. The pearl oyster is placed in a shallow tray of seawater. When the oyster begins to open, a wedge is placed between the valves. A piece of undamaged mantle tissue from another oyster is cut into a small square and is inserted into the gonad, along with a piece of shell, the nucleus, which has been machined to a rough spear. The size and shape of the nucleus determines the size and shape of the pearl used per oyster. If a half pearl is desired, the nuclei are placed between the mantle and the shell valve. After this operation, the oysters are placed in a cage covered by a wire frame. Cages are suspended from rafts in calm waters while the oysters recover from the operation. After about 6 weeks, they are inspected and the dead oysters are removed. The oysters are finally suspended from rafts or long lines for 3 to 4 years, being raised periodically, cleaned, encrusting organisms. The waters that produce the best pearls are often not good for growth. Therefore, it is common to start the culture out in a place where the oyster grow well and then transfer them to an area where the pearl quality will improve. In successful operations, the graft tissue grows over the nuclei and forms pearl sacs. The epithelial tissue secretes a nacreous or pearly substance that is deposited on the nuclei. Next, coming to seaweed culture. Seaweeds are a marine algae are primitive plants and they constitute one of the commercially important marine living resources. They grow in the littoral and sublittoral regions up to 20 to 25 meters depth in the sea and also in the estuaries and backwater areas. They belong to four groups, namely green, brown, red, blue-green algae based on the kind of pigments present in them and morphological and anatomical characters. The photochemicals such as agar, carrageenum, and sodium alginate are extracted from the seaweeds and they are used as gelling, stabilizing, and thickening agents in food, confectionery, pharmaceutical, dairy, textile, paper, paint, and varnish industries. Many protein-rich seaweeds such as alwa, entomorpha, caulerpa, codium, and monostronta, that is green algae, sargassum, hydroclathrus, laminaria, undaria, macrocystis, porphyra, gracilaria, Euchima, Laurentia, and Acanthophora, that is red algae, are used for human consumption in the form of soups, salads, curries, etc. Jelly, jam, chocolate, pickles, and wafers can be prepared from certain seaweeds. Marine algae are also utilized in different parts of the world as animal feed drugs and fertilizers for land crops as they contain more than 60 trace elements. Carbohydrate, iodine, bromine, vitamin, and certain antibiotic substances. About 700 species of marine algae have been replaced along the east and west coasts of India. It is estimated that the total standing crop of all seaweeds in Indian waters is more than 1 lakh tons, consisting of 6,000 tons wet weight of agar yielding seaweeds and 16,000 tons wet weight of algin yielding seaweeds. Now, seaweed ecology. Since seaweeds are attracted to a substrate by a holdfast and need light for photosynthesis, they are found only in shallow coastal waters of less than 200 meters, but are common only in about the first 35 meters of water. The geographical distributions of various species of seaweeds are largely related to the temperature, light, and tides. The vertical distribution of the seaweeds, that is, the depth at which they are found, has been explained only recently. For a long time, scientists claimed that, generally speaking, green seaweeds were found at the shallowest depths, red seaweeds were dominant at the deepest depths, and brown seaweeds were found at intermediate depths. It was assumed that because of the way that sweeter filters out light of different wavelengths, this distribution of seaweeds was related to the types of chlorophyll and accessory pigments characteristically found in each phyla. New experiments and statistical analysis have shown that there is no difference in depths of these seaweed groups. Now coming to reproduction, the life cycles of many species of seaweeds are rather complex. Reproduction may be vegetative or it can be sexual, involving the fusion of sex gametes. Many larger species of seaweeds go through an alternation of generations, similar to mosses and ferns. They may be represented in the sporophyte phase, which is diploid or the haploid gametophyte stage. The size, duration, and shape of the stages will vary with the species. What are the important seaweeds? Sugar rack, laminaria is cultured widely in the Orient. Natural populations of laminaria are limited to cool water. The saprophyte begins to deteriorate at the temperatures above uh, 60 degrees centigrade. Undaria is a brown algae grown on stones at depth of 7 to 15 meters in estuaries in cool waters of Japan, China, and Korea. The most popularly cultured seaweed is the red algae, Porphyra in Japan and Europe. There are about 25 species, most of which are found in the Pacific Ocean. The nori porphyra sheets are wrapped around rice to make a favorite Japan dish called sushi. Now we have come to the end of this chapter. Let us just discuss the summary. Prawns are the primary cultivated organisms in coastal agriculture, aquaculture system. Milkfish, grey mullet, mullets, and bhetki are the species suitable for brackish water culture. Organized culture of bivalve mollusks is the priority area of mariculture in India. The tiger prawn is the largest growing species among pinnate prawns. The culture of tiger prawn consists of pond preparation, stocking, and pond management. Pond preparation involves site selection, drying, liming, watering, and manuring of pond. Stocking and post-stocking and pond management involves procurement of seed, acclimatization, stocking, and water management, supplementary feeding, and finally, harvesting and marketing. Tiger prawns are also cultured in freshwater, freshwater ponds with saline soils. There are four types of culture systems in shrimp, depending on stocking during water management and feeding. Example, traditional, extensive, semi-intensive, and intensive. Mariculture not only includes production of food but also culture of other than food items like pearls, which are of expensive value. A pearl consists of concentric layers of nacre or matter of pearl. There are four types of pearls: regular pearls, irregular pearls, semicircular pearls, and reflectory pearls. Most of our pearls are obtained from pearl oysters. Freshwater pearls producing mussels belong to the family Unionidae. The process for pearl culture involves four stages, that is, collection of mussel and preoperative culture, surgery, post-operative culture, harvesting. Pond culture period of operated by bivalves depends on the fertility of water. So with this, we have come to the end of this chapter. Let us see the frequently asked questions from this chapter. Answer the following questions in about 30 lines each. Write about tiger shrimp pond preparation. Write about tiger shrimp stocking, post-stocking pond management. Write in detail about oyster culture. Write in detail about pearl producing mollusks. Write in detail about freshwater pearl culture in the world, in India and in India. Describe about the mantle tissue insertion method. Write in detail about gonadal insertion in pearl culture. Write about seaweed culture. Now, these are some more questions. Answer the following in about 10 lines each. These are the short questions. Procurement of seed for tiger shrimp culture. Stocking for tiger shrimp culture. Manuring for tiger shrimp culture. Supplementary feeding for tiger shrimp culture. Harvesting and marketing of tiger shrimp culture. Culture of tiger prawns in freshwater. Write about prospects of oyster culture. Write about post-spat collection period for oyster culture. Write about types of pearls. Write about pearl producing mollusks. Write about mantle cavity insertion in pearl culture. Write about marine pearl culture. Write about seaweed ecology. Then write about seaweed culture in sea. Hope you have understood the chapter, students. Thank you.